Bismillahirrahmanirrahim. This is the next lecture of the series. After gametogenesis, learning the formation of the female gamete and the male gamete. The next lecture definitely comes the union of the male and the female gamete known as fertilization. In today's lecture, we'll learn the definition of fertilization. We'll learn what are the prerequisites for fertilization. We'll also learn what is the site of fertilization, and we'll learn also what are the events of fertilization, and at the end, the achievements and the abnormalities of fertilization. There is a comprehensive lecture on fertilization and the related events. Let's go. First of all, we should learn the female organs. That is the vagina. This is the uterus. These are the fallopian tubes on either side. And these are the ovaries, here and here. Okay. Next, briefly, the sperms are deposited over here during sexual intercourse at the upper part of the vagina. From here, the sperms, they ascend up through the cervix, through the cavity of the uterus, then toward this side or that side, through the fallopian tube to its distal dilated end known as the ampulla. The distal part of the ampulla has finger-like projections which hang over the surface of the ovaries. From here, the female gamete is coming out, and here the male gamete comes, and they here unite together. The process known as the fertilization. We'll see. So, how we define fertilization? It is a fusion or union of male and female gametes. When the sperm's chromosomes, they combine with those of the mature oocytes to form a fertilized egg known as the zygote. So the simplest or the very brief would be the fusion of the male and the female gametes to form a zygote is known as the fertilization. It would be enough if you remember this thing. A few important things you must remember before the fertilization to discuss. That is the oocyte is viable for 12 to 24 hours after ovulation. Meaning thereby, when the oocyte is released from the surface of the ovary, if it is utilized for the union of the male, gamete, that is the sperm, within 12 to 24 hours, a product which we desire, the formation of the zygote, we can achieve. After that, the oocyte may be there, but it is not capable of union with the male gamete or with the sperm. On the other hand, the sperm is viable. Viable means capable of union with the female gamete. So the sperm is viable for 24 to 48 hours after ejaculation. So when it is out of the male genital tract, the sperm is capable of union with the oocyte within 24 to 48 hours. After that, it is not capable of union and not able to achieve fertilization. For fertilization to occur, sexual intercourse, technically called coitus. We call it coitus, the union of the husband and the wife in privacy when they come closer together. The sexual intercourse means the entry of the male organ, that is the penis, into the vagina of the female. That is the sexual intercourse. And this intercourse causes the ejaculation of the sperm into the vagina. For fertilization to occur, this sexual intercourse that must occur not more than 2 days before the ovulation or 24 hours after ovulation. So this is the time framework in which the fertilization is possible. After ejaculation, meaning thereby after the sperms are out of the penis, they are deposited in the vagina. The accompanying prostatic secretions, they cause the semen to be clotted as a curd-like structure, not a fluid thing. But within minutes, this liquefies. And much of it leaks out of the vagina outside, in the perineum. Acidic vaginal environment destroys sperm. The vaginal epithelium causes secretions, and that secretions, if they are acidic, they will destroy the sperms. Actively motile sperms, they will make way through the cervix to the uterus. The sperms, if you remember, the structure of the sperm, it has got a head, it has got a neck, it has a long tail. So tail is used for the mobility of the sperm or the movement of the sperm from the site of deposition of the semen in the vagina to move up through the cervix into the uterus and above through the fallopian tubes. So another thing to be remembered is not only the tail movement of the sperm propels it upwards through the uterus, but the peristaltic contractions, which are reversed from lower to the upper side, from the cervix towards the fundus or towards the upper part of the uterus, and then towards the fallopian tube's distal end. So the reversal of the or backward movement of the contractions of these organs, that also helps to ascend the sperm from the upper part of the vagina to the site of fertilization in the distal part of the fallopian tube. So at the time of ejaculation, about 3.5 milliliter of semen is deposited in the vagina. And each milliliter contains about 100 million sperms. So roughly, at the time of coitus, 350 million sperm are deposited in the vagina. But only 300 to 500 arrive at the site of fertilization. That is the ampullary region of the uterine tube. And only one of them enters the oocyte to fulfill fertilization. All the rest, they are wasted. No more needed. So you can see how much, with immense population of the sperms, the male Almighty Allah has provided. Another thing is noted that 7 hours are mandatory in the female genital tract for the sperms to stay before fertilization. This is called the capacitation of the sperm. And during this time, the protective coats on the surface of the sperms, they are evaded or removed by the secretions of the uterus, etc. So it is 7 hours before a sperm is able to fertilize the female egg to stay in the female genital tract. That time is known as that event is known as the capacitation of the sperm. Now let's move on. Events of fertilization, what are those? In this picture, this is the vagina, sorry, this is the uterus. That is the vagina lower down where the sperms are deposited. From here, they are moving up to the cavity of the uterus, from the body. Then they pass on to the fallopian tube or the oviduct, and its distal part known as the ampulla. This is the ovary, and this is the Griffin follicle, which ruptures, and the secondary oocyte and the first polar body surrounded by zona pellucida and a layer of corona radiata cells that is out. These finger-like projections of the distal end of the fallopian tube, they move a sweeping movement towards the lumen of the oviduct or the fallopian tube. So they drag it into the cavity of the fallopian tube. This structure is now here in the fallopian tube. So what is out of the ovary at the time of ovulation? That is the secondary oocyte, this one. And along it is the first polar body after the completion of meiosis 1. Outside is the glycoprotein coat known as the zona pellucida. And on the most outer side would be a few layers of the granulosa cells or the follicular cells all round. This is known as the corona radiata. This is the simplest way of designing or making the secondary oocyte after ovulation. What you see over here is that is the lower down. This one is the secondary oocyte. This is the zona pellucida. These are follicular cells or the granulosa cells, these ones. And here you see the sperms moving towards the secondary oocyte. And see over here, this is the nucleus of the sperm. And on the nucleus, there is a cap-like structure of acrosome, a structure filled with hydrolytic enzymes. Okay? Acrosomal reaction, the enzymes are out, they digest to make a hole in the zona pellucida, and after that the sperm contents, they enter into the wall, through the wall of the, or through the plasma membrane of the oocyte, and finally enter into the cytoplasm of the oocyte. Okay, let's see. Another view. This is zona pellucida. This one is the zona pellucida, and here is the secondary oocyte. Along the cytoplasm outside, near the plasma membrane of the oocyte, you see these small globules. These are lysosomes, which will cause a reaction. We'll just see. Here again, the sperm, its head, its neck, its tail. The sperm has got a vesicle or a cap-like structure full of enzymes known as the acrosomal vesicle. Okay. This layer would unite with the plasma membrane of the sperm head, and from here small holes will be created, and through the holes the enzymes would come out. They will digest a hole in the zona pellucida, and thereby this sperm would pass through the zona pellucida, and when it has passed, it will come in contact. It's this layer or the inner layer of the acrosomal cap, which is now forming basically the outer layer of the sperm head, that will make contact with the plasma membrane of the oocyte, that would dissolve, and the contents of the sperm, they would all go inside. Okay, and simultaneously these cortical granules, which are in fact lysosomes of the secondary oocyte, they would release their contents out in the pericellular space over here, extra the space between the zona pellucida, and the plasma membrane of the secondary oocyte, and they would create a reaction. We'll see that is known as the cortical reaction. The implication of that reaction we'll see. So this is the another simplified view to understand the events of fertilization.
Excuse me, sir.
जी.
Excuse
जी.
me, sir. Repeat कर दे पिछली diagram.
This is the zona pellucida.
Sir, enzyme का name क्या है?
आप पहले बात तो सुन लें जो आपने पहले सवाल की. This is zona pellucida. This is the secondary oocyte. These are the sperms. And these are the follicular cells through which they are making way to get contact with the zona pellucida. Now here the acrosomal caps release enzymes which are hydrolytic enzymes, hyaluronidase mainly, which digests the zona pellucida and make a way for the contact of the sperm head with the plasma membrane of the secondary oocyte. Finally, this layer of the sperm head would make contact over here and would dissolve, and all the contents of the sperm, they would be in. They would get inside. And at the same time, these cortical granules, which are the lysosomes of the secondary oocyte, they would release their contents over here, and they will make something which we'll discuss in the next slides. We're coming down to understand the events of fertilization. The first event is the penetration of the corona radiata. Second is the penetration of the zona pellucida. And finally, third is the penetration of the secondary oocyte cell membrane. So these are in fact the barriers through which the sperm is to pass before it enters into the cytoplasm of the secondary oocyte. So one by one, penetration of the corona radiata. So one thing is important. Only motile sperms are able to disperse the granulosa cells of corona radiata. Meaning thereby, the sperms must be active and live. If it is not so, that cannot disperse the granulosa cells. It is simply a physical reaction or physical movement through the tail of the sperm, which by undulating or waving movement or propelling movements, that creates a space between the granulosa cells. So the motile sperms, they disperse the granulosa cells of the corona radiata by propelling and waving movements by the tails to reach the zona pellucida. As soon as the sperm head, they touch the zona pellucida, acrosomal reaction sets off. The acrosomal reaction begins. And what is that acrosomal reaction? The outer acrosomal membrane fuses at tiny spots with the sperm head cell membrane, and the points of fusion, they rupture, and releasing the acrosomal contents or acrosomal enzymes, which are hydrolytic, hyaluronidase basically. They digest holes, or they digest the parts of the zona pellucida, creating holes in the zona pellucida. So acrosomal reaction is the release of the acrosomal enzymes which digest holes in the zona pellucida. Once the first sperm head touches the secondary oocyte cell membrane, the cortical granules, which we just saw, these are basically the lysosomes of the oocyte. They release their enzymes, and what that enzyme is, zonal inhibiting proteins, ZIPs. These send back a quick wave of depolarization of the oocyte cell membrane, preventing further sperm fusion with the oocyte cell membrane. One. Secondly, they destroy the sperm receptors. And what is the result? That the already bound sperms, they get detached. So this is the mechanism by which not more than one sperm contents can enter into the cytoplasm of the secondary oocyte. So the cortical reaction is basically prevention for the penetration of the further sperms into the cytoplasm of the secondary oocyte. Now comes the third barrier, that is the plasma membrane of the oocyte. After acrosomal reaction, inner acrosomal membrane practically acts as the plasma cell membrane. It fuses with the oocyte cell membrane. Fusion area dissolves. All the sperm contents, except the cell membrane, they enter the oocyte cytoplasm. All the sperm contents, meaning thereby the nucleus, the mitochondria, and other contents, the contents of the tail, microtubules, they all enter into the cytoplasm of the secondary oocyte. Immediately the oocyte completes meiosis II. Okay? I'm speaking not oocyte, but secondary oocyte. So immediately, when the sperm contents, they have entered the cytoplasm, the secondary oocyte would complete meiosis II to form a mature oocyte, which would be having 23 chromosomes and single structured chromosomes and one nDNA, and the second polar body. So the secondary oocyte immediately completes meiosis II, forming a mature oocyte and second polar body. The nucleus of the sperm decondenses. Decondenses means when the spermiogenesis last time we discussed, we noticed the rounded nucleus of the sperm assumes a pyramid-shaped structure by condensation. The nucleus of the sperm decondenses, it swells to form the male pronucleus. Rest of all sperm contents, they degenerate. What is left behind? Only the nucleus. The nucleus of the mature oocyte also swell, and they form the female pronucleus. So the male pronucleus by the sperm head and the female pronucleus by the oocyte, by the mature oocyte's nucleus. Now the male and the female pronuclei, they fuse. The nuclear membranes rupture. Now the 23 chromosomes of the sperm combined with the 23 chromosomes of the mature oocyte to form a fertilized egg known as the zygote. And this is the final stage or the accomplishment of fertilization. So what do we achieve after fertilization? What are the achievements? Restoration of the diploid chromosome number. So 46 chromosome number is achieved. Determination of the sex, male or female. Beginning of development of a new individual. So the moment the fertilization is complete, the next stage, that is the cleavage, would begin without any interruption. You cannot stop. So the beginning of the development of the new individual ushers the moment the fertilization is complete. So these are the achievements of the fertilization. Now associated, there are some abnormalities of fertilization. Infertility, sterility, superfetation, superfecundation, and parthenogenesis. So what is infertility? Can anybody speak?
Sir, inability to fertilize.
Anyone else? Anyone else? So infertility is not inability. It is failure. Some disease of the male or some disease of the female is inhibiting fertilization. If you remove or treat that disease, the couple is able to get a baby. So this is infertility, which is treatable, which is cured. Like there is pelvic inflammatory disease of the female. If you treat that, the fertilization is possible and next steps of development of the baby is possible. Similarly, if there are less sperms in the female, in the male, if you give some treatment, then it is possible that the sperm count is more or sperm count is restored, so the fertility is possible. Infertility is basically failure to conceive, failure to become pregnant of a female because of disease of herself or because of disease of her partner. Sterility is in fact the inability. Here, it is a permanent problem. You cannot get fertilization because of the problems of the male or the female. If, say, the lady has got uterus, but it is not properly developed, so you cannot have fertilization process complete. If the ovaries, they are not functioning, you cannot get fertilization. Similarly, if the male testes are not, they are but there, if there but not functioning, you cannot get it. So sterility is a permanent disability. It is inability to achieve fertilization due to the congenital diseases of the male or the female partner.
सर जो स्टेरिलिटी है उसको हम इन विट्रो फर्टिलाइजेशन से क्योर कर सकते हैं?
नहीं कर सकते, क्योंकि उसमें आप उसमें जो डिसेबिलिटी है वो कंप्लीट है। आजकल उसमें वो वजाइना नहीं कुदरत ने बनाई,
ठीक है,
उसके अंदर यूट्रस नहीं बनाई गई, उसके अंदर एग्स नहीं बनने वाले दोनों ओवरीज। ये परमानेंट डिसेबिलिटीज, congenital absence, you cannot get it. Okay. Superfetation. Normally, during pregnancy, females do not ovulate. So Almighty Allah stops ovulation during pregnancy. But sometimes one of the ovaries may ovulate. And if it is that the time is so that the male and the female, they come closer, they meet in privacy, so that can also get fertilized. So a fetus is there, another fetus also comes following the formation of the previous fetus. So this is superfetation, meaning a fetus over the fetus. Another fetus is coming out, which is very, very rare, very, very uncommon. Superfecundation. This is also very, very uncommon. It is that the relationship of a pregnant woman with when other non-genuine male partner, if that happens, there are two babies coming down, one having one father, the other having the other father, which is also very, very uncommon. But last year or before that, I will just share the picture in the next slide. It was noticed in Vietnam that the twins, they are there, one having one father, the other having the other father. Okay. Parthenogenesis. Parthenogenesis is what? Fertilization is necessary for the development of a new individual. Without fertilization, a new baby cannot. But it has been noticed in the lower species that electric stimulation or other stimulation can start development of the new organisms. This is known as the parthenogenesis without proper fertilization. If development can begin, we will call it as parthenogenesis, which is not in human.
खुदा दा वास्ता साडी जान छाड़ दियो कुछ जेडी 4 दिन जिंदगी बची है ना ਅਸੀਂ ਤੁਹਾਨੂੰ ਬੜਾ ਕਿ ਤੁਹਾਨੂੰ ਬਦਮਾਵਾਂ ਦੇ ਵਿੱਚ ਕਿ ਤੁਹਾਡੀ ਜਾਨ ਕੱਟ ਛੱਡੀ
Sir, parthenogenesis please repeat कर दें.
See that? This is a quiz recorded in March 10, 2016. You can go to the BBC site and read it out. This was noticed in Vietnam.
Okay.
So we have reached the end of the lecture. शिकवाए जुल्मते शब से तो कहीं बेहतर था अपने हिस्से की कोई शमा जला दे. Thank you very much.